Hey guys, it's Miss Adelini. I'm going to be creating 3 videos for you today. One about the history of DNA, one about the structure of the DNA molecule, and one about DNA replication, how it makes a copy of itself. Okay. Back in the day, scientists thought that protein was the cell's hereditary material, the molecule that got passed down from parents to offspring. And they thought that because proteins are extremely complex, and they thought they needed a complex molecule to determine complex hereditary traits. Number 1 in your packet says early scientists believed that blank was the genetic material of the cell. Please fill in the term protein and explain why based on my explanation. Proteins are made up of 20 different amino acids, which are the building blocks of proteins, and they can be combined many different ways. And those amino acids are linked together in chains called polypeptides. Number 2 in your packet should be amino acids. Number 3 should be polypeptides. And you may want to draw an arrow from number 3 to the image on the top right-hand corner of your page, because that is a polypeptide. Now, there were several scientists who did experiments to determine that there was a molecule that determined hereditary information in the cell, and that molecule was DNA, not protein. The first scientist to conduct experiments on this was Frederick Griffith, and he worked with pneumonia bacteria. In his first experiment on the left-hand side, he used a type of pneumonia bacteria that was harmless to mice. He injected the mice with those bacteria, and oh hey, they lived. Then, in his second experiment, he injected mice with a dangerous strain of that pneumonia bacteria. They got pneumonia, and they died. So thus far, we've just had 2 control groups to show us what harmless bacteria do and dangerous bacteria do in the mice. Then he wanted to see if he could kill off the dangerous bacteria by heating them up. So he heat killed our dangerous strain in his third experiment, and then those bacteria were unable to make the mouse sick. He wanted to see if he mixed our harmless strain, our little blue dots, with the heat-killed dangerous bacteria, if some sort of genetic material could be passed from the heat-killed dangerous bacteria to the harmless bacteria to make them dangerous. So what he did was he mixed those 2 types of bacteria, and he found that the mouse did get infected with pneumonia bacteria and did die. So, another term for those dangerous bacteria would be virulent bacteria, and he called those his S bacteria. Another term for the harmless bacteria would be non-virulent bacteria, and he also called that his R strain. And ultimately, in his 4th and final experiment, he found that he could take the R strain and make it harmful when it took in some of the DNA or genetic material from his heat-killed, dangerous, harmful bacteria. So, his study suggested that there was some genetic material passing from the heat-killed, harmful bacteria to the R strain bacteria. So, let's go back to your packet. We're on number 4. Frederick Griffith worked with pneumonia bacteria. And number 5 asks, what did he find to be true after his experiments with the S and R strains of bacteria? And you're going to go with the middle bullet point here. He found that the R strain, and you might want to write in parentheses, harmless, could become virulent when it took in DNA from the heat-killed S strain. And maybe in parentheses next to S strain, you write harmful or dangerous. So that's number 5. Number 6 in your packet says Griffith's experiment proved that blank is the cell's genetic material. And the bottom bullet point tells you that our study suggests that DNA was probably the genetic material. This isn't completely accurate. Griffith really didn't figure out that DNA was the molecule being passed from our S strain to our R strain bacteria. It was another scientist later on. He just figured out that some molecule was being passed. Okay. Our second experiment was conducted by scientists Hershey and Chase, and they worked with viruses. And I'm going to give you a little information about viruses. They're not like normal cells. They're not considered cells because all they are is a coat made of proteins, shown here, and genetic material or DNA on the inside. This right here is a type of virus called a bacteriophage, and that's a type of virus that can only infect bacteria cells, so that wouldn't be harmful to you. Okay. So Hershey and Chase weren't sure whether it was the protein coat from the virus or the DNA on the inside of that protein coat that got injected into the host cell. They knew that whatever material the virus injected into the host cell was going to be that virus's genetic material. Since the virus injects its genetic material into the host cell so that the host cell can make copies of the virus. It's their way of reproduction. So, Hershey and Chase wanted to figure out whether it was the protein coat or the DNA that got injected into the host cell. So they did two experiments. In the first one, they radioactively labeled the protein coat so that they could see it. And they allowed the virus, the bacteriophage, to infect the host cell, the bacteria. What they found was that the radioactivity stayed outside the bacterial cell, indicating that it was not being injected into the host cell. So they concluded that protein probably wasn't the genetic material of the virus. They did the same experiment again, but instead of radioactively labeling the protein coat, they radioactively labeled the DNA. When they allowed that virus to infect the bacteria, they saw that the radioactive label ended up inside the host cell. That shows us that DNA is probably the genetic material of the virus. So number 7 in your packet asks, how did Hershey and Chase's work with viruses help to support this idea? And their experiment showed that DNA was the genetic material of viruses. All right. Next scientist we're going to talk about is a woman named Rosalind Franklin. And Rosalind Franklin took X-ray pictures of DNA. And they're called X-ray diffraction pictures. Diffraction means to bounce off. So they were using X-rays bouncing off of the DNA molecule and analyzing the pattern that was created on the screen. The picture that she came up with is this one up in the top right-hand corner of the slide. Now, to me, that doesn't look like anything really. To the trained eye, someone who's experienced at analyzing X-ray diffraction photographs, that looks like this molecule at the bottom right-hand corner, which is the DNA double helix. Watson and Crick were able to analyze her photo and create the first model of DNA, which is a double helix, using her images. So we've answered number 8 and 9. Rosalind Franklin took our photographs, and number 9, Watson and Crick built the first model of DNA, and you're going to fill in double helix into the last blank on number 9. All right, guys, that's it for DNA history, and our next video is going to focus on the structure of DNA.